This video about genetic roots of humans aging and why we live so short. Please, you can download presentation, the link to the presentation in the description to this video. And the video is relatively long, so it will save time, and also you will get access to the sources and to additional information, because you can click on links in the description and in the PDF file, and you can get more information about this. So, what we will talk about? The fossil record has always contradicted evolution theory. Instead, it contains data predicted based on the species creation theory. Darwin was aware of this, but anticipated that future generations would discover the necessary evidence of species evolution. This has not occurred. Thus, we may consider as a point of discussion that new biological species were created, while evolution theory explains only how the species adapted to the changing climate and environment. The mechanism of species creation remains a mystery. Also, so-called intelligent design theory explains many biological facts. Unfortunately, mainstream science refuses to accept it. It is mathematically proven that evolution theory does not explain the creation of new biological species. For example, evolution theory has no explanation for the so-called Cambrian explosion, when a large number of new biological species appeared without any evidence of common ancestor. Evolution theory has been proposed long before the discovery of genes and the structure of the chromosome. No wonder that evolution theory cannot explain the fact that DNA represents an intelligently organized and functional digital code. This is a schematic presentation of the Cambrian explosion, when so many new forms of living organism and new species appeared without common ancestor. Additionally, evolution theory is unable to explain the enormous complexity of living cells and their organelles, as well as complexity of multicellular organisms. We know that animals of different species with different number of chromosomes cannot usually interbreed and produce fertile offspring. Horses, for example, have 64 chromosomes, while donkey have 62 chromosomes. And mule is the offspring of a donkey stallion, and a mare, a female horse. A hiney is formed by a male horse and a female donkey. Mules and hinnies both have an odd number of chromosomes, 63. So this is a hinny. It is like small horse, and mule with head reminding the donkey. The offspring is incapable of producing gametes, which such as eggs and sperm. However, this has been about 60 recorded anecdotal cases of fertile female mules since the 15th century. Okay, you can click on the link and get the more information on it. Another example of fertile hybrids was mentioned in another publication. You also can get access to it. The domestic horse has 64 chromosomes, as I said, and while the Przevalski horse has 66. Nevertheless, the 65 chromosome hybrids are fertile. Interesting, yeah? On the contrary, tigers and lions both have 38 chromosomes and may produce viable hybrid offsprings. But female hybrids are completely fertile. Male hybrids, on the other hand, are sterile. Okay, you see that again different species can interbreed. Okay, and even they have different number of chromosomes, or the same number of chromosomes, but the result of the offspring can be fertile or infertile. Okay, here we see pictures of that representing interbreeding between the tiger female and lion male. This is the so-called liger male, and the tiger male and the lion female produce Tygon male. And that's you see that what is again interesting that, as I said, female hybrids are completely fertile. Male hybrids are sterile. Okay, so these hybrids are sterile, infertile. During the meiosis cell cycle in infertile hybrids, genes from two different species are swapped. The result is a hybrid chromosome with a unique combination of genes in gametes, ensuring that the genetic information from the hybrid parents is combined. This swapping of chromosomal segments happens in the interphase and prophase 1 of the meiosis cell cycle during the process of producing gametes. The exchange of chromosomal segments between non-sister chromatids is referred to as crossing over. During crossing over, a portion of the father's chromosome is swapped for the portion of mother chromosomes. Here it is schematically represented, this meiosis and crossing over, exchange of the fragments of the chromosomes, and this is explain how genes can be transferred from one biological species to another biological species if the offspring is fertile. Humans are fundamentally different from primates in their physiology, and social behavior. It is usual for primates to live in harems, which are groups of females led by alpha male. As a result, all other beta males are unable to have offspring. Furthermore, primates' menstruation cycle lasts about a year. As a consequence, the period of sexual desire, estrus, occurs only once a year. In contrast, women, humans, have their periods every 4 weeks, and sexual arousal might occur multiple times a day. Therefore, most humans live in monogamous families rather than harems. So this is a typical harem with alpha male and a group of females with offspring. And in difference, the human family is mostly monogamous with one man, one female, and children, common children. Humans differ from primates also in that they can cooperate, live in tribes, feel empathy for other tribe members, and even establish independent national states. It is already proven that direct predecessors of modern humans were Cro-Magnons. However, modern humans most likely inherited some genes from Neanderthals. It is only 1.5 to 2.1% of genome in general. Some individuals have more or less. And then, as I said, number of these genes varies between human individuals. This can be explained in part through crossbreeding between Cro-Magnons, Neanderthals, and Denisovans. Science has traced evolution of Neanderthals as biological species for about 500,000 years, while Cro-Magnon people mysteriously appeared about 45,000–50,000 years ago. The Cro-Magnon people were anatomically similar to modern Europeans, and according to the reconstruction of Cro-Magnon skulls and skeletons, their phenotypes correspond to our perception of individual physical beauty. Neanderthals had larger brain, broader features, larger eyes, more prominent brow ridges, and larger teeth than Cro-Magnons. Okay, so here's the schematic presentation of the Neanderthal man, Cro-Magnon, and hybrid of Neanderthals and Cro-Magnon as is our modern man. Neanderthals were rushless and efficient predators who hunted even at night and ate any animal. The Neanderthals' daily diet was nearly 5 kilos of meat. They, like apes, lived in family groups of 10 to 30 individuals, and Cro-Magnons lived in larger tribe-like groups of up to 100 individuals and had a highly evolved social system involving trade, art, and sophisticated rituals. Neanderthals and Denisovans may be subspecies of the same species. They went extinct about 40,000 years ago, probably because they were unable to compete for food resources with Cro-Magnons. Differences in appearance, social organization, and genetic code suggest that Neanderthals and Cro-Magnons were distinct biological species. It is not yet clear whether Neanderthals had 46 chromosomes like humans or 48 like apes. However, as mentioned above, crossbreeding and gene transfer can occur even between species with different number of chromosomes. Modern humans do not have Neanderthal Y or Y chromosomes and Neanderthalian mitochondria in our cells. It is possible that fertile offspring of Neanderthal men and Cro-Magnon women were daughters, or otherwise the only fertile offspring of Cro-Magnon males and Neanderthal women were boys. So two possibilities how it can happen. According to Wikipedia, Neanderthals probably had the same number of chromosomes as modern humans, that is 46. Also, there is a hypothesis of an end-to-end fusion of two small ape-like chromosomes named 2A, 2B to produce human chromosome 2. See article in the Wikipedia. A real fusion that occasionally occur in nature have very specific features. And the overwhelming scientific evidence indicates that the fusion never happened in human chromosome, never. Okay, this is, you can read it is very long article, and in details it is proven that fusion cannot be, it cannot happen. Indeed, in case of fusion, we would see two active centromeres in the fused chromosomes and a long fused telomere site. Such chromosomes would be dysfunctional and cell will not live. Contrary to many evolutionary paleoanthropologists, claims that fragmented hominid fossil records does not show that humans gradually evolved from ape-like hominids. Again, evolution theory doesn't work. It has it is not proven by fossils. Also, humans are distinguished from hominids by unique moral, intellectual, and creative capabilities that cannot evolve gradually. Only so-called intelligent design theory, which was mentioned before, can tentatively explain the emergence of Cro-Magnons. However, modern humans are not descended solely from Cro-Magnons. Rather, modern humans are hybrids of Cro-Magnons and Neanderthals, as I said before. We may find indication of crossbreeding of created humans, which were probably Cro-Magnons, and some hominids, which are Neanderthals, in the Bible. According to the Bible, Adam and Eve were created after other animals were created. So we may claim that Neanderthals existed long before Adam and Eve were created, and due to science it is 500,000 years, and Cro-Magnons appeared only 50,000 years ago. According to the Bible, Cain, the first son of Adam and Eve, was cursed by God and exiled to the land east of Eden for murdering his brother Abel. There he met and married a humanoid woman from whom he had a son named Enoch. It is possible that humanoid creatures were Neanderthals, this is how I explain it, and the only fertile offspring of the Cro-Magnon and male and Neanderthal woman were boys. Yeah, this is how the Bible talking about, so it is exactly can be explained by science. Otherwise, we must acknowledge that Cain's wife was his sister, which is highly unlikely. The individual presence of or absence of some important genes obtained from Neanderthals, in particular, indicated by the rhesus factor of the blood. A negative rhesus factor indicates the absence of specific genes from primates. Next, all chimpanzees have a type A blood, all gorillas have type B blood, and Cro-Magnons had O-type blood. It is also interesting that Neanderthals were carriers of a gene that prevents the digestion of milk or lactose, sugar in milk, in adulthood. There is little known fact that humans with rhesus negative blood live on average longer than ones with rhesus positive blood and can digest milk well into old age. The blood of such humans has a more alkaline reaction and they get infected less frequently. Also, in difference from Neanderthals, Denisovans, and other primates, almost all present-day humans carry a serine to glycine amino acid substitution in the gene of a critical enzyme, which is glutathione reductase. So you can go check name and see the publication. So the human version of the enzyme reduces oxidative stress and may contribute to greater longevity of humans in comparison to primates and Neanderthals. Neanderthals had early puberty development and early aging of the body, while Cro-Magnons are characterized by late puberty development and later aging of the body. Accordingly, on average, Neanderthals lived only about 22 years on average, and Cro-Magnons lived over 50 years but also suffered from high mortality in harsh environments. Therefore, as a working hypothesis, I might suggest the possibility of a gradual shortening of the lifespan of biblical patriarchs by the accumulation of the Neanderthal genes in their genome due to cross-breeding of the species. Also, shortening the lifespan of animals, why Neanderthals live short, can be explained, and shortening of lifespan can be a side effect of evolutionary adaptation of mammals to living in harsh environment. In particular, quick healing of wounds is necessary to survive in the situation of frequent injuries, accidents, and skirmishes with other members of the same biological species. Stages of the quick wound healing include local inflammation, activation of fibroblasts to produce collagen, local stiffening of the extracellular matrix, and transition of some fibroblasts to senescent cells if extracellular matrix stiffness exceeds some threshold level. Senescent cells then produce pro-inflammatory signal molecules SASP, senescent associated secretory phenotype, to attract macrophages. Macrophages consume debris, dead and senescent cells to convert them into nutrients for stem cells. Stem cells replace damaged and dead cells to complete healing of the wound. However, slow stiffening of extracellular matrix due to glycation reactions lead to accumulation in time of the senescent cells and chronic inflammation of all tissues. Meanwhile, stiffening of blood vessels leads to hypertension, chronic hypoxia, and oxidative stress in tissues. In turn, chronic oxidative stress leads to degeneration of healthy cells into cancer cells, accumulation of lipofuscin inside and outside cells. Interesting that senescent cells can be eradicated in condition of intermittent hypoxia. For example, naked mole rats experience frequent cases of this intermittent hypoxia. They don't accumulate senescent cells and don't show stiffening of extracellular matrix or blood vessels. Therefore, they actually don't show signs of senile degeneration and live 10-20 times longer than other rodents of compatible size. Humans cannot change own genetics but can simulate conditions of intermittent hypoxia by regular high-intensity interval training or breath-holding exercise. Otherwise, intermittent intake of senolytics can be practiced. The elimination of lipofuscin and other debris from the extracellular matrix could be the next step in radical lifespan extension. I believe it can be done with super enzymes that can convert lipofuscins and oxidized lipoproteins to nutrients in an acidic environment. Also, humans don't produce such enzymes. Some bacteria do. It it is yet unknown how these enzymes can be efficiently transferred from the colon to the macrophage lysosomes. Again, I believe I have some progress in this direction, but again, it is probably can be improved. Okay, calcium oxalate is another harmful waste which accumulates in many tissues, including our brain. So in conclusion, I believe that with supplements, a healthy diet, and regular exercise, people can slow down and even reverse the aging process. Okay, so my idea of this video is such that humans live short because of interbreeding, because they are hybrids of Cro-Magnons and Cro-Magnons and Neanderthals. So, and plus in time, this evolution adaptation to quick wound healing was probably also very important for shortening of lifespan, and also the diet that humans started to consume too much sugar, too much carbohydrates, and reaction of sugars with proteins create stiffening of extracellular matrix, and then all this cycle of chronic inflammation and accumulation of senescent cells go, and then our life is much shorter. So with proper diet, we can improve it, and again, regular exercise to get this intermittent hypoxia is also important, or taking senolytics. Okay, then I believe that we are on a good way, and it may work. That not only aging could be slowed down, but it can be reversed. So thank you for taking time to watch the video. Please comment, subscribe, like, ask questions, and share information. And again, I tell you that all my project is to extend radically the lifespan of humans. I believe it is possible. Humans can live as long as biblical patriarchs, so up to almost to 1,000 years, if we will do all right, and if we will create conditions that all these genes which we got from Neanderthals will be suppressed. So, and it can be done with proper diet, exercises, etc., and supplements. So it can be done. We can live almost the same as long as Cro-Magnons, and I mean Cro-Magnons were living 50 years or a little bit longer, because the environment was not very friendly, and they were starving, and they were getting injured, and infections, etc., etc. But in good conditions, and if everything will be done properly, humans can extend lifespan to a few hundred years, for sure. Okay, have a wonderful day. Ciao, ciao.